Morning, everyone. I hope everyone's having a good spring and enjoying this nice weather. Today, it's my pleasure to introduce Dr. Linda Baughn. Linda has had just a remarkable career, and she's still a young person and has even more to accomplish. But Linda grew up in Ohio, and after getting a bachelor's degree at Miami of Ohio, she moved to Tufts, where she got her PhD under the mentorship of Naomi Rosenberg. After that, she did two prestigious postdocs. The first was at Weill Cornell with Selena Chen Kiang. Then she moved to Albert Einstein, where she worked with Maddie Scharf. So I first met Dr. Baughn when we both kind of started back at the University of Minnesota. Linda came to the University of Minnesota within the Department of Genetic Cell Biology and Development, where she was an assistant research professor. And she built the lab and started doing research, and we were introduced through Dr. Brian Van Ness and immediately became collaborators and friends. After a few years of working at the University of Minnesota, she made a transition to do a combined cytogenetics and molecular fellowship, 2 years with Doctors Hirsch and Dolan and 1 year with Barat and Dr. Yohi. And after that 3 year program, she was recruited to the Mayo Clinic, where she's just had this incredibly remarkable career. So Linda went to the Mayo Clinic, and there she initially started uh, working in their cytogenetics and fish and microarray areas and really became a leader clinically. But she was very quickly able to gain traction and gain independent grants, first through the Mayo Clinic and externally. And what's happened over the course of her career is that she's had such a breakthrough in her scientific and clinical expertise that she was awarded more than $2 million from the NIH to get a sizable R01 so that she can spend more than 80% of her time doing research. She's published more than 120 papers in her lifetime, the majority of them at the Mayo Clinic, and I'm sure she probably has another 10 in submission right now. She was a chapter author on more than two of the World Health Organization chapters, including the plasma cell myeloma, and she's built up this really neat program. So I've had Linda speak once before externally for a different organization that I'm involved in. But one of the really cool things about it is that um, in addition to her really incredible clinical acumen, she's developed this wonderful research program looking at the biology of translocation 1114 and myeloma. And I know you guys are going to say, oh, you're just excited because it's myeloma. But I guarantee when you hear Dr. Baughn present, you're going to be really excited to see this mechanistic insight that she's discovered and the story that she's going to tell today. So Linda, I'm so proud to have you speak with us today. I can't even tell you how proud I am of all your accomplishments, and I really, really greatly appreciate you as a colleague and a friend, and I look forward to your presentation.
Thank you. That's actually a really sweet introduction. Mike and I have been friends for a while, and I'm really grateful for his collaboration and friendship. And it's also hard to believe that it's been almost 10 years since I was at the U, so time has gone by really fast. So I assume everyone can see my screen. So today I'm going to talk about 1114 myeloma, and I just have one disclosure. So as I'm sure you're aware, multiple myeloma is a malignancy of long-lived post-germinal center plasma cells. And from a very simplistic perspective, they basically fail to undergo apoptosis and maintain their self-renewing capabilities. So this aspect is going to come up later throughout the whole talk. So again, I'm sure the audience is aware that for multiple myeloma, there are basically two main genetic subtypes. Again, from a simplistic perspective, one is about half of the cases have an immunoglobulin heavy chain translocation, and the most common partner gene here is the CCND1 gene, and this is found in about 18 to 20% of myeloma. There are other recurring IgH translocations, including FGFR3, NSD2, as well as others. And the other half have a hyperdiploidy, which is characterized by trisomies of odd-numbered chromosomes. So a question that I have been interested in asking, and even when I was in Maddie Scharf's lab at Einstein, was how do these primary IgH translocations occur in the first place? And it is fairly well understood that they occur within the germinal center due to the normal process of affinity maturation. So within the germinal center reaction, you have the enzyme activation-induced cytidine deaminase, which is important for somatic hypermutation and class switch recombination. So somatic hypermutation purpose is to initiate point mutations within the VDJ segment of the immunoglobulin heavy chain gene. And this basically enables infinite repertoire of antibodies that can interact with different antigens. And class switch recombination is important for switching from making, for example, an IgM antibody to IgG, IgA, IgE. So this is a normal process that happens, but it is thought that for unknown reasons, when things go awry, you can have the immunoglobulin translocations to other partner genes. And how do we know this? One reason that we can say this is because looking at the breakpoints, for example, an 1114 rearrangement, again in the Sergos plot, just focusing on cyclin D1 and the immunoglobulin heavy chain gene with the VDJ and the constant regions, you can appreciate that most of these breakpoints are actually located within the constant region or in the region that is between the constant region and the IGHJ region. So this suggests that these are probably due to AID-mediated somatic hypermutation, as well as possibly class switch recombination, because most of these breakpoints are actually found within the switch regions. So I've been interested in 1114, and I'll tell you the reason why. One of the reasons is that it actually is fairly unique in its biology in comparison to other immunoglobulin heavy chain rearrangements in myeloma. It is known that this subtype is more likely to have a small mature plasma cell morphology, and it retains B cell-like expression features, including the expression of CD20 and PAX5, which are typically expressed more in mature B cells rather than plasma cells. The other interesting aspect is that it is the only subtype that is associated with a known germline risk allele. And this is actually a really interesting observation, and possibly one that can help us understand more about the mechanism of 1114. So the G allele of the SNP RS9344 has been associated with 1114 rearrangement. It was found by Niels Weinhold in 2013, and it is associated with both myeloma as well as the pre-malignant condition, monoclonal gammopathy of undetermined significance, with an odds ratio of 1.8 to 2. Interestingly, this G allele is not associated with increased risk for T1114 mantle cell lymphoma. So this probably tells us something about the mechanism, in that 1114 mantle cell is more likely due to aberrant VDJ recombination mediated by RAG1 and RAG2 within the pre-germinal center rather than a post-germinal center reaction. And also, it's interesting that the G allele is not associated with other immunoglobulin heavy chain translocations in myeloma. So the molecular mechanism for how this SNP, which is, I will comment, is a fairly common SNP, the mechanism has not been really well characterized until now. We also noted that this SNP has a very high risk allele frequency in Western Africa and South America. We were curious to know whether this could be possibly associated with increased risk for 1114 in populations that have high African ancestry. So we initially looked at a myeloma cohort of individuals, or I should say actually individuals that had an abnormal FISH result. So that could have been gut smoldering or multiple myeloma. And we specifically enriched this cohort for those of high African ancestry, and we noted that the G allele was significantly associated with individuals with over 80% African ancestry in comparison to individuals that had less than 0.1% African ancestry. We also looked to see whether this G allele was associated with increased prevalence of T1114 in association with African ancestry, and we were able to see this only when we showed with using a high cut point of African ancestry of over 80%. So using the risk allele in a multivariate model, we can show that it is highly associated with t(11;14 even after we adjust for age, sex, and race status. So it was really just the distribution of the risk allele and its association with t(11;14. So this led us to ask, really, what is the molecular mechanism behind why the G risk allele is associated with an increased risk for t(11;14 myeloma? So to do this study, we used multiple cohorts. One, we took advantage of the 1,000 genomes cohort as well as Roadmap Epigenomics Consortium because we were interested in looking at epigenetic associations between the risk allele in various immune populations. We utilized two different myeloma cohorts, the Multiple Myeloma Research Foundation COMPASS cohort, which includes about 700 patients with myeloma. And here, publicly available access to whole exome, whole genome, and RNA-seq is available. And we also had our own Mayo Clinic cohort of 661 patients with multiple myeloma, and we had available whole exome and RNA-seq. We also generated our own ChIP-seq and ATAC-seq data so that we can look at the epigenetic analyses in this way. We use generalized linear regression models as well as an interaction model. And we also perform cell line validation studies. So our first question was, we were curious whether the G allele was associated with 11;14 myeloma in these two independent cohorts, and whether the G allele can influence cyclin D1 expression within t(11;14 myeloma. We figured that this was probably the most likely scenario, but we looked at other possibilities as well. So within these two cohorts, we did confirm that the G allele of RS9344 was associated with t(11;14 in both the COMPASS and Mayo Clinic cohort with an odds ratio of 1.88 to 1.54. We were also interested to know whether there was a preference for the G allele in t(11;14 using patients that were heterozygous for the risk allele. So either having on one allele an A or on the other allele a G. And for this, we took advantage of the RNA-seq data. So as you would imagine, you would have an 11;14 event that would typically occur only on one of the alleles. And when we looked at the sequence of the expressed cyclin D1 gene from the RNA-seq data, we could determine on which allele the translocation occurred on based on whether we had a G expressed or an A expressed. So it was actually fairly binary in that we know that most of these plasma cells are not expressing cyclin D1 unless there is an actual translocation. And so it was extremely clear-cut and very simple to do this analysis. And what we found was that of the 117 heterozygous cases, 80% of them had the G allele expressed from their cyclin D1, suggesting that it was the G allele that was selected as the allele involved in the 11;14 event. And this demonstrated a fourfold increased preference for the 11;14 translocation to occur in cis with the G allele in comparison to the A allele. So as you would expect, there is, as we would certainly expect, an increase in cyclin D1 expression in association with 11;14 in comparison to non-11;14. And since I mentioned that the G allele is associated with the t(11;14 event, in a univariate analysis, we were able to confirm that the G allele was positively associated with cyclin D1 expression. But the real question actually is the G allele associated with increased cyclin D1 expression within the t(11;14 cases? And so using the generalized linear regression model, we could show that the G allele actually only influences cyclin D1 expression when there was an 11;14 event, not when there was an 11;14. Without an 11;14 event, the G allele does not influence cyclin D1 expression. So this positive association of the G allele on cyclin D1 expression was driven by the interaction of the SNP with 11;14. So in order to have the effect, we needed to have the 11;14 event to occur. And we calculated, or we estimated, the interaction effect from the G allele accounting for 20% of cyclin D1 expression. The majority of cyclin D1 expression was driven by the IGH, the repositioning of the enhancer sequences from IGH, and 20% were coming from the G allele. So the question we asked was, how can the G allele influence cyclin D1 expression within 1114? Are there differences in chromatin accessibility and histone modifications that occur within cyclin D1 in the context of 1114? And does the G allele influence these differences in chromatin accessibility? So first, we wanted to understand this locus further. So again, RS9344 SNP is located within cyclin D1. And there were 11 variants in LD with the SNP in individuals of European ancestry. This covered a region of 12.3 kb within the cyclin D1 gene. And these were all located within the same TADs from Hi-C data from the GM12878 cell line. So this cell line is actually really valuable in that it's a EBV transformed B cell line that we have been using in our analysis. And all of these SNPs were located within the same geographical location. So we further then looked at whether there were differences in histone marks in association with different transcriptional states within this locus. So as a reminder, these various histone marks tell us something about whether the region is, for example, a poised enhancer or an active enhancer. So we looked both at 3 different marks: H3K4 trimethylation, H3K27 acetylation, and H3K4 monomethylation. If there was only the presence of H3K27 trimethylation, it would be considered a repressive mark. And when we have all 3 of those marks active, it would be considered an active enhancer region. And when there was the presence of both the H3K27 trimethylation and monomethylation and trimethylation of H3K4, then that would be considered a poised mark. So we looked at, again, we took advantage of the healthy immune samples that were publicly available, as well as our own t(11;14) myeloma samples that we had generated our ATAC-seq and ChIP-seq data on. So we identified 6 regulatory regions that were covering 22 of the 23 SNPs. So I mentioned actually that there are 23 SNPs with a lower LD with RS9344. So we actually used all of those SNPs in this analysis. So these were the 6 different regions that were identified within the cyclin D1 gene region. And what you can appreciate is really just comparing the profile from the healthy samples to the myeloma samples. There is a fairly big difference in the profile. So in the healthy samples, we do see an enrichment of H3K4 monomethylation and H3K27 trimethylation in the regions 1 through 3. But you can see that the H3K27 acetylation is essentially absent in these regions. So this suggested to us that this region is a poised enhancer, but if you compare that to the t(11;14) samples, you see an enrichment of H3K4 trimethylation, H3K27 acetylation, and H3K4 monomethylation in this entire region, regions 2 to region 6 in t(11;14), suggesting that this region is functioning as an active enhancer. So when you compare this region now from the t(11;14) cases to non-t(11;14) myeloma, you actually see that the non-t(11;14) myeloma region looks very similar to those of healthy samples. And so this suggests that following the translocation of the immunoglobulin enhancer regions to cyclin D1, there is a very significant increase in chromatin accessibility and also an increase in enhancer activities. So we looked at this region further in the patient samples as well as in 2 different myeloma cell lines. And we were able to determine that cyclin D1 itself was actually considered a super enhancer within this region. So several years ago, actually, our collaborator had published a study that showed that actually just the translocation of, for example, an enhancer region near a proto-oncogene such as cyclin D1, it itself is associated with a epigenetic spreading of these what they called these broad domain of H3K4 trimethylation, which by definition is associated with high H3K27 acetylation and H3K4 trimethylation. So it's not just that you have this enhancer sequence and you're getting increased expression of cyclin D1, you're actually getting the spreading of these histone marks into the cyclin D1 region. So next we wanted to ask, actually, so big deal. Is the G allele actually associated with increased active histone marks? So to do this, we took advantage of the cases that were heterozygote. So where on one allele they had an A and on the other they had the G. I already mentioned to you that you're more likely to get the 1114 event to occur when you have a G allele. And so using the ChIP-seq and ATAC-seq data, it shouldn't come as a surprise to you that when we look at whether the G or the A allele was expressed in the sequence reads that we were able to pull down, were more likely to be G allele. So we look for this allelic imbalance. So almost all of the sequences had the G allele. But what we really want to ask actually is that if you have a translocation that does happen to involve the A allele, is the G allele still associated with an increase in these histone marks? So I mentioned to you that 80% of the time you're going to have the translocation on the G allele, but 20% of the time you're going to have it on the A allele. So we had to hunt and hunt for cases that were 1114 positive that actually had the translocation on the A allele. So you can imagine that it's actually pretty hard because we had to actually have the whole exome sequencing data too to know in the RNA-seq data to know which allele the translocation was involved in. So of the few cases that we were able to find, you can see that we have an increase in H3K27 acetylation in association with the G allele rather than the A allele. So even if the A allele is involved in the translocation, it's associated with reduced H3K27 acetylation. So we wanted to actually study this even further. So we took advantage of the KMS12PE cell line, which is actually, interestingly, has the translocation on the A allele. So we CRISPR edited the cell line so that it actually has the G allele, and we identified two independent lines, and we performed the same ChIP-seq and ATAC-seq studies, and as well as qPCR for cyclin D1 expression, and we also have some single-cell RNA-seq data as well. And when we did the CRISPR edit, you can see that there was an increase in H3K27 acetylation in association with the G allele in comparison to the A allele, the wild type, the A allele. So here we have quantified this observation, and by qPCR, you see that this is associated with increased cyclin D1 expression and also increased cyclin D1 expression by single-cell RNA-seq for a different project that I'm going to mention later on. So the next question we asked was, are there differences in the recruitment of transcription factors to the cyclin D1 enhancer region that could influence cyclin D1 expression beyond what the effect is of the IgH enhancer? And also, in order for this to be interesting, we would have to actually identify differences in transcription factor binding recruitment in association with the G allele. So we took advantage of the ENCODE data where we, again, going back to the GM12878 cell line, this cell line happens to be heterozygous for the SNP. So one allele has the A and the other allele has the G. And In the ENCODE data, they have available ChIP-seq data for 80 different transcription factors. And we looked specifically at the cyclin D1 region, and we were able to identify 3 transcription factors that showed a binding peak within this region, including the RS9344. And we chose to focus on PAX5 for evaluating this analysis further. And one of the reasons why was because while we were working on this paper, there was another lab in Calgary, the Bayless lab, which identified, interestingly, that the t(11;14) subtype actually is able to have an increase in PAX5 footprint. So these data came from ATAC-seq data, where they looked to see using the known PAX5 binding motif, they were able to show that there was an increase in accessibility of the predicted PAX5 binding motifs, specifically within 11;14 in comparison to non-11;14 cases. And this is probably due to the fact that PAX5 is more likely to be expressed in 11;14 compared to non-11;14. But these data excited us in that it showed that PAX5 may actually be functional within the 11;14 subtypes. So with that knowledge, we looked to see whether our SNP was located close to a PAX5 binding motif. And sure enough, it was one base pair from the RS9344 SNP. And again, going back to the concept that the GM12878 cell line is heterozygote for the SNP, we were able to show that there was preferential binding to the G allele relative to the A allele. So again, using the ChIP-seq data for PAX5, if PAX5 showed equal binding to this region, independent of G or A, we would have had 50% of the reads having a G allele or 50% having an A allele. But here we're seeing a preferential binding of the G allele. So this suggested the possibility that the G allele could be associated with increased binding of PAX5 to within the cyclin D1 region. So we wanted to test this hypothesis. And first, what I'll show you is that within both of the cohorts of myeloma, the 11;14 subtype was associated with increased PAX5 expression in comparison to non-11;14. And as we would expect, we have an increase in the cyclin D1 expression in association with the G allele versus the A allele. And when we incorporated into our generalized linear regression model, the level of PAX5 expression, whether there was a G allele or an A allele, and whether there was an 11;14 event or not, we found that in combination, the highest cyclin D1 expression was associated with elevated PAX5, the presence of the G allele, and t(11;14 status. So this suggested to us that there was an interaction between these variables with creating the highest cyclin D1 expression. So we wanted to test this hypothesis further, and we utilized the U266 cell line, which is actually not a t(11;14 rearrangement, but it actually has an insertion of the alpha enhancer sequence upstream of cyclin D1. This cell line is also heterozygous, and we know that this enhancer is inserted into the allele that has the G allele here. And we know this because this is the data from the genomic DNA. We see both A and G indicated here, but when we perform Sanger sequencing from the expressed allele, you can see that you only have the G allele present here. So when we overexpress PAX5 in the U266 cell line, you see an increase in both PAX5 expression by qPCR as well as the protein, and we also see an increase in cyclin D1 expression. So this indicates that adding more PAX5 is associated with increased cyclin D1 expression. So this slide basically summarizes a lot of negative data that we also generated. We were curious to know, after the TLM14 event actually occurs, do you have a difference in the distribution of somatic mutations? So the 1114 event is necessary, but it's not sufficient for myeloma pathogenesis. So we were curious to know whether the G or the A allele, 1114 cases, would have a different trajectory to their development of myeloma. But what we really found was no significant difference in the acquisition of somatic mutations. We were also curious to know whether there was a difference in the translocation breakpoints between these cases, and we also were not able to see that. We had hypothesized whether there was potentially a difference in where the breakpoints were occurring, whether, as we know, mantle cell lymphoma, they're more likely to occur closer to the cyclin D1 gene in comparison to myeloma, which is spread a little bit further, and we didn't really see a difference. We also looked at whether there was a distribution of a different TLM14 expression subtypes. And these two expression subtypes are termed CD1 and CD2. And we were unable to find a difference here. And we also didn't see a difference in the overall myeloma survival. One major limitation of these studies is that, as I mentioned to you, the sample size for the A allele is very small. So of the 1114 cases, only 20% of them will have the translocation on the A allele. And so I think that it's possible with time, we can acquire many more whole genome sequence data from those of the A allele, and maybe we will see a difference, but nothing was obvious for this sample size that we had here. So in summary for this portion of the talk, I hope I have convinced you that TLM14 does correlate with increased expression of both cyclin D1 and PAX5. The G allele in combination with IgH is associated with increased cyclin D1 expression via PAX5 within the TLM14 cases. We believe that the higher cyclin D1 expression mediated by the G allele is happening after the 1114 event, and this may create a selective advantage for the plasma cell to undergo malignant transformation. I do think that there's a lot more probably to the story. There are probably other genomic events that could contribute to this observation, and we hope that we can actually understand this even better. Okay, so going back to TLM14 and its unique biology. I'm sure everyone is aware that TLM14 has increased expression of the pro-survival protein BCL2, and it also has lower expression of the anti-apoptotic MCL1 and BCL-XL. So the increased expression of BCL2 has been associated with the increased susceptibility of TLM14 to BCL2 inhibition, primarily through the drug venetoclax. So we established a collaboration with multiple investigators to try to understand the biological mechanisms of venetoclax response. Because so far venetoclax is not FDA approved for TLM14 myeloma. So I'm only going to present a portion of the story, one that is associated with cyclin D1, but I will mention to you that here we had 44 whole genome cases, sequences, and 24 whole exome data sets from 33 TLM14 patients from both Rochester and Arizona. 10 patients had whole genome sequencing from pre-venetoclax and post-venetoclax. So the main, I guess, punchline of the story is that we identified that the presence of RAS mutations had the strongest association with outcomes. So if a patient had a RAS mutation, it was strongly associated with shorter progression-free survival and reduced venetoclax response. But we were particularly curious about the observation of the paired samples. So following venetoclax relapse, we identified 20%. So the cohort is still very small, but 20% of the cases had a deletion of this gene called PMAIP1, which encodes for the pro-apoptotic NOXA protein. So we wanted to model this observation in using cell lines. So we went back to the same cell lines that I mentioned to you, the KMS12PE. These are 1114 positive, and we generated venetoclax resistant cell lines after continuous treatment in the lab with venetoclax over a period of 6 months. We also generated venetoclax resistance from our knock-in counterparts, mainly because we were curious to know, could there be something interesting about the risk allele status and its response to venetoclax and also acquisition of venetoclax resistance? So what you can appreciate is that all of the resistant cell lines had a reduction in the NOXA protein. So again, as a reminder, the PMAIP1 gene encodes for NOXA. But you can also appreciate that there were other changes that happened in these cell lines in association with these anti-apoptotic and pro-apoptotic proteins. So we collaborated with a laboratory at Emory University, and they had generated in the same cell line PMAIP1 knockout, and they showed that in these 1114 cell lines, when you knock out the PMAIP1 gene, you actually show very significant resistance to venetoclax. So this demonstrated that this gene is important in the response to venetoclax. So we focused on this gene because of those observations, but also because going back to the cohorts that I described before, we were able to show a very clear association between high PMAIP1 expression and t(11;14) status. In fact, there was a significant positive association between PMAIP1 and cyclin D1 expression. So we wanted to study this further, and we created a cyclin D1 knockout U266 cell line. We actually tried to knock out cyclin D1 from 3 different myeloma cell lines and 3 mantle cell lymphoma cell lines, and the only one where we could actually knock out cyclin D1 was the U266 cell line, and it's most likely because this happens to be an RB1 null cell line, so it's less reliant on cyclin D1 for its cycling capabilities. So in this, we identified 2 different clones. So here our strategy was, again, this is in concert with Centigo, who performed the knockout. So an A base was inserted into exon 1 of the cyclin D1 gene, and this resulted in a stop codon in exon 2. So interestingly, even though we're not actually able to get cyclin D1 expression, protein expression, we do get some transcript that is generated, but there's obviously nonsense-mediated decay happening. But what's interesting about this genomic region is that even though we don't have any cyclin D1 protein, we actually retain the enhancer activity of this region based on our ChIP-seq data. So this kind of keeps the enhancer region active, but we're not getting any protein. I will note that in this cell line, we actually do get fairly significant reduction in cell cycle still, but the cells are able to retain some cell cycle capabilities. So in this knockout cell line, you can appreciate that we have essentially no NOXA expression, PMAIP1 expression, and we're also not getting any protein, demonstrating that the knockout of cyclin D1 was associated with a complete loss of PMAIP1 expression. So going back to the knock-in cell lines, you would imagine that if you do have an increase in cyclin D1 expression, you may have an increase in PMAP1 expression. And we were able to show that in both of these cell lines here, where we have an increase in the gene expression by qPCR, and we also have an increase in the protein NOXA. We were also able to show that the elevated PMAIP1 expression is associated with increased response to venetoclax. And we were also curious to know what is potentially the mechanism behind why cyclin D1 is associated with PMAIP1 expression. And we turn to our H3K27 acetylation data, and you can see that the knock-in lines were associated with increased H3K27 acetylation in comparison to the wild type, and the knockouts had essentially no H3K27 acetylation. So this leads us to kind of the bigger question that we have is, what is the role for cyclin D1 in transcriptional coactivation? We do know from other studies, not in myeloma, that show that cyclin D1 can actually activate transcription by recruiting transcriptional coactivator histone acetyltransferases, CBP. And what is also very fascinating to me is that cyclin D1 most likely can actually have multiple roles besides simply its role for interacting with CDK4 and CDK6 and mediating the cell cycle. The reason why this is interesting is because it's a little bit counterintuitive in that if you have the t(11;14) event occurring within a germinal center B cell, so that cell most likely is cycling, but when it undergoes the differentiation toward the plasma cell stage, those cells actually do not cycle. And so I don't believe that the main function of cyclin D1 in that context is just for the cell cycle capabilities, because those cells are just not cycling. So there's probably a much broader role for cyclin D1, and it may be related to its ability to activate transcription, potentially through its the mechanism of modulating the histone modifications. So we do know from the knockout cell lines that we have very significant differences in gene expression as well as H3K27 acetylation on a global level. So this leads me to the second summary. This second part was a little bit shorter, but we demonstrated that loss of PMAIP1 or NOXA appears to be a mechanism for venetoclax resistance in patient cells and in cell lines. And we were able to show that cyclin D1 is associated with increased PMAIP1 expression via modification of H3K27 acetylation. So our current studies are focused on evaluating how cyclin D1 plays a role in promoting H3K27 acetylation and gene expression, not just at the PMAIP1 locus, but at other loci. And I think that really our long-term goal is to understand the true function of cyclin D1 in promoting myeloma pathogenesis and disease progression beyond the cell cycle. So I thank you very much for your attention. I do want to acknowledge my research group, these are the members who have done essentially all of the work that I presented, as well as the previous members. I want to acknowledge the Mayo myeloma group, who have been very instrumental, particularly in allowing me to obtain patient samples at Mayo. Our collaborators on this specific project, Francesco Mara and Marcela Cadora, the collaborators at Emory University, Larry Boyes and Vikas Gupta, our population geneticist, Iran Alhaik, and our collaborator, Anita Miklasova. And I definitely want to acknowledge my funding. And I actually didn't talk about the primary goal of our laboratory is really to understand the molecular basis for why individuals of African descent are more likely to get multiple myeloma. We have some really interesting projects on focusing on the tumor microenvironment and drug response. So that's it. And if people have questions, I would love to be able to answer them.
Thank you, Dr. Baughn. It looks like Dr. Ade has a question.
Yes, I do. Excellent presentation, Dr. Baughn. And I have just so many questions, but I'll leave it to one or two. So I like the epidemiological information about the G allele in cyclin D1. That's really quite impressive. And, looking like it's really very clean in the West African and the South African community. But most of your study seems to be in the African population, right? So do we know anything about the role of this G allele and the T1114 translocation in the South American population? The reason why I'm asking is that in West African, there could be a lot of other factors that modify the importance of this allele, like malaria and things like that. So what do we know about the South American? Is it the same thing? And then I have a second question if I have time later.
Yeah. No, I think it's a really good question. I have to say, I feel as I continue working on this project, I feel like I understand less and less about the role of, like population genetics and the G allele. I don't know. I think to answer your question, we certainly need to have additional samples from South America. We have to understand all of the environmental aspects. I don't think that it's as simple as I kind of lay out. I think that it's probably, there's so many different environmental effects. And one thing that I've just been thinking recently, even if we have an increased frequency of the SNP, we do know that the SNP specifically is associated with increased risk for 1114 in this specific population, but we don't know whether it's like a certain baseline level and it has to go, we have to have a further increase in the G allele distribution. We just need to increase our cohort diversity, and we actually, I think, have to repeat all of the PRS calculations on all of these different diverse populations and take into consideration the environmental component. So I don't know if I've actually answered your question.
No. I suspected that we probably don't know much about this, but I think it's just going to be very important because there's some other mutations in the West African that we know, are driven by, infections. We have the buckets and you have all those ones. So it will just be, interesting to know that. I have a second question before Dr. Nelson, if you may. And that's probably a little bit related to my first question. So this going outside of lymphoma right now. So in my part of the neck of the woods, in the GI, and then some breast and everything, there are other mechanisms for cyclin D1 mutation that's not from rearrangement with, by translocation. So do we know, and I'm not sure whether we know the answer to that, but does this polymorphism, this SNP, does it really impact any of these mutations, like point mutations in cyclin D1 and all the other mechanisms? Do we know? Or is this something we should be studying?
Yeah, so I can only talk about what we know in the context of myeloma. So point mutations in cyclin D1 are not very common in the context of myeloma. They're actually, myeloma, I would say, is primarily a malignancy of structural variants. But we did look to see if there were differences in the acquisition of mutations, primarily because our initial hypothesis was that the G allele in myeloma must be somehow associated with increased AID activity, because we're not seeing that increase, just the increased G allele in mantle cell lymphoma. And unfortunately, we did not see a significant difference in cyclin D1 mutations. We even looked to see, could we have, mutations within AID hotspots? We were not able to identify any kind of difference. And that work was done by Francesco Mara, and he has a massive amount of data on that. So unfortunately, no, I think that our hypothesis was not correct initially regarding potentially increased AID activity. Again, we thought, like, could the breakpoints be more likely to be within AID hotspots? But again, we couldn't find anything that would argue for a specific mechanism. So that's a really good question. But for like breast cancer or other solid tumors, I don't know. But in an odd way, paradoxically, and if you look at some of those other studies with the risk allele, they actually talk about the A as being the risk allele for some of these other studies. So, and even I think Karen Knutson had generated a mouse model where she had shown that the A allele was more pathogenic in her, I think it was a breast cancer model, mouse model, but I don't remember exactly, but I know that the A allele was. And one thing I didn't mention here, because we had a lot of negative data, right? And so with those other studies, they believe that the reason why the A allele is maybe more pathogenic in their context without like immunoglobulin heavy chain translocation is because it actually is a splice. It creates a leaky splice site, meaning that if you have an A allele, you are more likely to read through into intron 4. And that, they think, is because when you do that, you generate a truncated cyclin D1 protein that lacks the nuclear export signal. And so the cyclin D1 is basically stuck in the nucleus in their model. I will say that we never found this alternative cyclin D1. We did show the leaky splice. So we do have a splice defect in our cell line and even in the patient sample, but by mass spec, we could not find the protein. And so it's possible the protein exists, we just couldn't detect it, but we don't feel that the splicing probably has a lot to do with the mechanism, but it could be in other contexts that it does. So I can only talk about what we see in myeloma.
Yeah, thank you very much. I really enjoyed yours. And I can see behind Mike Linden that there's a whole lot of cyclin D1 in the picture. Anyway, thank you.
Dr. Nelson.
I was going to say, Linda, or you're right, and the protein doesn't exist, and going after the transcriptional read-through is just not relevant.
Yeah.
It's good to see you. Thanks for a great talk, and congrats on all the success. It's been awesome to hear about that. I'm going to go back to Deli's first question, and I was trying to think about this too. So you have really, really nice data showing a connection between the G allele and levels of cyclin D1 expression. And in my mind, at least, and you can tell me if you disagree, but in my mind, that basically that creates a setup that when you've got that allele, you have the translocation, you're going to drive higher levels. It's essentially a setup for an evolutionary selection that's going to be a more oncogenic mechanism. I'm wondering though if there are other things about the G allele haplotype that might also change the genomic context that actually predisposes to the translocation. Like, have you done Hi-C or any other types of technologies for genomic context, potentially in some of these populations that have a higher MAF of the G allele, looking at the AA versus the AG versus the GG?
Yeah, we tried. So I reached out to a collaborator from someone at Einstein, who I work with at Einstein, and he tried to in silico model. So basically, like, the purpose of his work was to model kind of like the genomic architecture to try to predict like increase AID activity.
Yeah.
He was not successful. The other, and I have to say, tragically, he passed away like really young. So I don't know why I'm mentioning that, but that did not work out. And the other thing is we established the collaboration with Anita Mikulasova because they had done, so, high C is like really hard to do, right?
Yeah.
So they established a model. So they modeled their high C using the GM12878 cell line, because there's like actual like wet lab high C data generated, and they then use the U266 cell line. And I had asked them to add in, like, to switch out the A and the G. And so they are going to be working on that, but we were not able to get that completed yet. I do think you're probably right. And actually, if you're interested, I can send you some papers on like the modeling that they did, where they show basically how flexible the cyclin D1 genomic region is and how it's interacting with like that MYEOV gene that's like really close to cyclin D1. And so I wouldn't be surprised if it could potentially change the genomic architecture in that region, but we just don't have any proof yet.
Cool. But it sounds like you're actually working on it, so that's really cool. I'm glad that it makes sense as a question. I'd love to see it. And yeah, we've tried high C and ovarian cancers, and boy, is that a nightmare in terms of what those plots look like. Because yeah, you take an HRD deficient tumor and you do high C on, and it looks like someone took a shotgun blast at the contact map.
Yeah. And I mean, the other question I guess we had was ChIP-seq using cyclin D1. So we would love to be able to show, like, and other people have shown it in mantle cell lymphoma, that when they do ChIP-seq, that they can actually identify cyclin D1 interacting with the DNA. We attempted ChIP-seq. Unfortunately, the same antibody that lab used is no longer available. So we tried 3 different antibodies, and we were not actually able to get the ChIP-seq to work. It's possible that cyclin D1 just doesn't interact strongly enough with the DNA, or it's going through a different, like another, like a transcription factor or some other mechanism. But it would be really interesting to see if it's sitting on the DNA or through something.
Cool. Well, thank you. It's good to hear all your work. You're doing a fantastic stuff.
It's good to see you.
And I realized in my introduction that I forgot to mention that Dr. Baughn quickly ascended to full professor. So, are there any other questions for Dr. Baughn? Well, great. I thank you everyone for your attention, and I hope you enjoy the rest of your beautiful May morning.
Yeah, thank you guys, everyone. It's really wonderful to be back.